Greetings, everyone. Today, we are delighted to be back with yet another exciting video dedicated to the fascinating and ever-evolving field of eye development. Our enthusiasm knows no bounds as we embark on this journey of exploration into the intricacies and wonders that surround the formation and maturation of the human eye. Through this captivating visual medium, we aim to delve deep into the scientific marvels and cutting-edge research that shed light on the remarkable processes shaping the eyesight of human beings. By delving into the biological, anatomical, and physiological aspects of eye development, we hope to enlighten our viewers with a comprehensive understanding of how this complex sensory organ takes shape from its early stages to its fully functional state. In our previous video, we discussed important points that the formation of the neural tube from a flat sheet of ectoderm is called neurulation. Initially, simple tube will develop and form the brain and spinal cord, and is the source of neural crest cells and their derivatives. Moving forward, this is ectoderm, this is mesoderm, this is endoderm, and this is notochord. This diagram shows early neurulation. Now, in next step, signals from the notochord start off the processes of neurulation in the ectoderm. In the next step, ectoderm involutes starts to form a tube. In the next step, neural crest cells appear in the crests of the waves of the ectoderm that are moving towards each other. In the next step, the neural tube has formed, and neural crest cells move away. Now consider this neural tube. From neurulation, the central nervous system continues to develop as the cranial end of the neural tube dilates and folds to form spaces that will become the brain. The remainder of the neural tube, caudal to the first four somite, will become the spinal cord. Neural crest cells have different destination. Now, let's understand the development of the brain. 3 vesicles develop in the cranial end of the neural tube. At day 28, the vesicles are the prosencephalon that become forebrain, mesencephalon that becomes midbrain, and rhombencephalon that becomes hindbrain. Now, in the 5th week, the 3 vesicles become 5. Let's understand it. Prosencephalon split into 2. Telecephalon and diencephalon. Telecephalon will become cerebrum. Diencephalon will become thalamus and hypothalamus. And this is mesencephalon. It remains same. That becomes midbrain. Rhombencephalon split into 2. Metencephalon and the myelencephalon. This metencephalon becomes the pons. Myelencephalon becomes the medulla, while this is spinal cord. But in this video, you need not to remember all these. Let's just focus on diencephalon. So, at day 22, when the length of the embryo is 2 mm, and when there are 8 somite, the somite are a set of bilaterally paired blocks of paraxial mesoderm that form in the embryonic stage of somatogenesis along the head-to-tail axis in segmented animals. This is forebrain. This is the surface ectoderm. Blebs from the forebrain form optic grooves. In the next step, this blebs from the forebrain form optic grooves that become optic vesicles. Then, in next step, this optic vesicles come into contact with the overlying ectoderm. It will trigger 2 things. They trigger the initiation of the lens placode, which is the thickening of ectoderm, and optic placode, while this is the optic stalk. Do you know which regulatory gene is involved in development of eye? Well, paired box 6 is involved in development of eye. In the preceding video, we embarked on an intriguing journey exploring the captivating process of the development of the eye from the forebrain. Delving into the intricacies of embryonic development, we witnessed the remarkable transformation of a simple cluster of cells into a complex and sophisticated organ, the eye. As we marveled at the gradual differentiation and specialization of tissues, we unraveled the essential steps that lead to the formation of the optic cup and lens, the pivotal components responsible for the eye's remarkable ability to perceive light and images. Now, in our forthcoming video, we eagerly anticipate delving even deeper into the wondrous realm of ocular development as we shine a spotlight on the fascinating intricacies of the optic cup and lens. Prepare to be enthralled as we dissect and elucidate the molecular and cellular mechanisms underlying their formation and functionality.
Welcome to sqadia.com, your one-stop destination for medical online learning. Dive into our extensive catalog of in-depth animated videos, meticulously tailored for students, residents, and even practicing healthcare professionals. With over 40 medical courses available, covering topics from the fundamental anatomy to the complex endocrinology and everything in between. Explore various teaching styles. Engage with our immersive videos, purposefully designed to make learning not only incredibly enjoyable, but exceptionally effective. To get started on your path to excellence, take advantage of our exclusive free trial and gain immediate access to a wealth of educational resources. sqadia.com, your journey to exceptional medical education begins here.